Okay. And we're recording. Okay, cool. All right, everybody. Okay, welcome back to the brief delay. So for some procedural announcements, right, we've got exam 2 coming up on Friday, and I will do a Zoom over Q&A just for my content, though, after class 11:15 to 12:15, if you want to do that. There is the first one of the quizzes will be after we finish this respiration and cardiovascular system content, which will be, I'll post it on Friday, and you'll have all of the break to do it. Finish it by Monday when you come back. Any questions about that? Okay, okay, let me just shrink that down. Okay, so today we're going to sort of continue on with our cardiovascular system, and we might get into some respiratory system. Last time we finished on this slide, which was sort of explaining why do we have a maximum heart rate, right? And it's really because the heart is, a muscle that requires relaxation to fill, and then contraction in order to expel that, right? And the amount of fluid that's going to get expelled for any contraction is equal to the stroke volume. Okay, so the stroke volume is something that is not absolutely. Excuse me. Let me add those people. Participants. Middle. Okay, cool. Okay, stroke volume is not something that's totally fixed. I mean, of course, it does matter how big you are, how big of a person you are. That's generally related to how big your heart is. Okay, as is your overall heart rate is generally going to be lower the bigger a person are. Okay? So stroke volume can be adjusted. How might it get adjusted? If your blood pressure is going up, say during exercise, and the blood flowing back into the heart is going to stretch the muscles a little bit more. Okay. So your stroke volume tends to increase as the amount of effort that you're putting into maybe an exercise or whatever work that you're doing increases. However, there is a limit, right? There's only so fast the blood can get back into the heart, okay? And if you beat too quickly, you will not allow that heart to fully, the atria and the ventricles to fully refill before squeezing it again. Thus, your body caps out the heart rate at some maximum level that sort of optimizes the amount of stroke volume relative to the number of beats per minute that you're making. Okay, so for example, right, so as our sort of power increases, our heart rate is going to continue to go up. The stroke volume starts to go up, and then at some point it turns over, it starts going back down at a particular level of power. And that means that the cardiac output actually stops increasing once you reach a particular heart rate. Okay. And so there's no sense in going any higher than that, because otherwise you're just going to be doing more. You're putting more effort on your heart, even though you're not actually achieving any better outcome. Okay. Now that maximum heart rate does vary with age. Okay, the older you get, the further down it goes. But that relationship, and people always are sometimes try to calculate what is my maximum heart rate from some equation. Okay, you can't do that. Okay, the equation describes the population really well, the population mean, but it does not describe any individual, because there's huge variations in people's genetics and people's sizes that is going to lead to a very large range of heart rates at a particular age. Okay, so as you can see here, right, this curve does go down, right? If you're 20 years old, your heart rate's going to tend to be significantly higher than if you're 40 or 50 years old or 70 years old, right? There's a continuous sort of linear decrease in heart rate. But at any given age, both in men and women, the range is huge. One standard deviation is about 11 beats per minute. So to capture 95% of the population, you have 45 BPM sort of range within that. So that just goes to show you that individual variations can be very important, even if you can describe the population by a particular equation. So that's heart rate. Now let's see what are the sort of pipes that these blood is getting pushed through. So if we look at this image, okay, these are two distinct blood vessels, and which one might be an artery and which one might be a vein? What sort of characteristics would you think would be consistent with an artery versus a vein? So this is a histology slide. You can see red blood cells here. And what you can see around here is that there's a very thick wall, right? That thick wall might provide sufficient elasticity to maintain a higher blood pressure, right? Here we have a very thinner and relaxed wall, a relaxed membrane around it. Okay, that's much more relaxed. That might be consistent with a much lower blood pressure, right? It's sort of like an elastic bag that can stretch out and then collapses back down when blood flows out of it. Okay, so the left side was an artery, and the right side was a vein. Now, if we look here at the overall physiology and anatomy of these structures, what we can see is that both of them have what's a cavity on the inside that's called the lumen. Okay, that is where the blood is going to flow through. Okay, then we have an endothelial layer. Okay, this is a thin layer of cells that's going to line that blood vessel on the inside. Okay, now that endothelium is present both in arteries, which are the big thick ones with thick walls, and the capillaries, which are incredibly thin, okay, and where all of the gas exchange is going to take place. And also they're in the veins. One feature of the endothelium is generally they're very smooth. Okay, so they're going to try to reduce the amount of friction for blood that's flowing through those materials. Now, in the capillaries, we have this endothelium, and we only have around the endothelium, we have a very thin layer of connective tissue just to hold it all together. It's almost like, tissue paper or something, okay, but even thinner than that. Okay, so that's called the basal lamina, okay? And why might we want to have the capillaries so much thinner in the walls than in the arteries and veins? Yeah. Diffusion. Exactly, more diffusion, right? Diffusion is only going to be an effective method of exchange on spatial scales of microns, okay? Maybe 10, 20 microns at most, okay? So we want to have the distance between where the blood is held and where the red blood cells are in this capillary to the surrounding fluids to be as thin as possible, okay? At locations where you want to do gas exchange. When you're in the major sort of plumbing of the system here, that's going to be having lots of fast bulk flow here of blood through it, like arteries. You don't care because you're not trying to, the arteries are not a site of exchange, okay? So they can be as thick as you want, okay? Now, one feature that let's see here. One feature that both arteries and veins have that capillaries don't have is that they have this sort of smooth muscle that then surrounds the endothelium. Now, the smooth muscle is thicker in arteries than in veins, but both of them have that. And that smooth muscle can contract or relax in a manner that will allow these tissues to get wider or narrower, which will then change the amount of resistance to flow down that particular blood vessel, as well as how fast fluid can flow through those blood vessels. Okay. Then on this sort of outside here, we've got a connective tissue, right? And this is sort of, again, just going to be like a wrapper around the entire sort of piece in order to to provide some elasticity. So in general, arteries are going to stretch, are going to have sort of more resistance to stretch. They still can stretch, but that resistance is going to allow a maintenance of pressure, of blood pressure during diastole, ventricular diastole, which is where the heart is relaxed. So when you have a ventricular systole, the heart is going to increase blood pressure, going to pump it out into the arteries, okay? Those arteries are going to stretch a little bit, and then sort of the energy that's stored from that stretching will slowly sort of push the blood back down, maintain pressure, and continue to push the blood through the systemic and pulmonary circulation. Okay, right. So one other thing that we notice here is that there's a linear flow here of blood from arteries into capillaries and then back into veins, right? So arteries are anything that's moving the blood away from the heart. Veins are returning material to the heart, and the capillaries are connecting these two systems. Now, the capillaries, however, the direction of flow is a little bit more complicated than them because they are really an incredibly sort of dense network of webbing, okay? So if you block like one little area of a capillary, it's going to be able to route around that and continue to have a blood flow in many sort of different directions. And the rate of speed of flowing in a capillary is going to be much slower than in arteries or veins, and we'll see why that is in a second. Okay, one of the places that has where are places where you might have really big capillary beds? Any place that's going to want to have a lot of exchange of material with the surrounding fluid. So that would be your muscles, because you're going to need that for exercise, but also places like the gut and the intestines. They're going to have very thick capillaries, very dense capillary networks that are going to be able to exchange nutrients that are getting extracted from digestion products. Now, when we think about flow of material in a pipe, okay, we have to consider both the pressure and the diameter of the pipe. Okay, so if you have a thin pipe, okay, let's imagine we have one long tube here, right? And we've pressurized one end of the tube, and we have less pressure on this end of the tube, so material is going to flow down the pressure gradient one end of the tube to the other end of the tube. However, if that tube has a variable diameter within it, the speed at which that material is going to flow through any individual slice of it is going to vary, right? Why is that? It's because there's more space for fluid to flow through this sort of wider tube than in this narrower tube, right? At any given slice of this sort of tube here, the same amount of material, the same amount of acid flow through that tube. So with the same, you're going to have an equal flux between this part and this part and this part and this part. So this is the law of continuity. So the law of continuity says that we're always going to have the same amount of material flowing through these different slices of tubes. But since the surface area here, and by surface area, I mean the overall surface area of what you're going to pass the material through, okay? So a narrow tube is going to have a surface area that's basically equal to the radius squared of the little circle here, right? That's going to be much smaller than this one, okay? So material is going to have to flow much faster through a narrow tube than through a thicker tube. And we can sort of quantify this overall flow rate. So flow here being Q, how much material is flowing through, is going to be equal to the pressure difference between one side and the other side divided by the overall resistance. So we're going to have low resistance here when you have a lot of surface area and high resistance here. So one confusing feature of this sort of explanation, though, is that capillaries are really thin, right? They're really skinny, and like the arteries are really thick, really wide. An artery has much less resistance to flow than a capillary. So why is it, if this law is true, why is it that blood is flowing much slower in a capillary than an artery? And that's shown here in this diagram. So if we look here and we look at the relative speed in centimeters per second of blood cells flowing through the arteries or through the aorta and then to arteries here and then to arterioles, we can see this flow slows down dramatically, okay, from sort of 0.5 meter per second down to basically moving at millimeters per second or even slower than that, okay? So very slow flow down here in the capillaries. So when you're considering how fast a blood vessel is going to flow, you need to think of not just the individual pipe that is going through, but the entire set of blood vessels that it could potentially flow through at that distance away from the heart. There's only one aorta. The aorta has a much higher diameter than any of these other blood vessels, okay? But there's only one of them, right? So the total surface area that is allowed that blood can flow through the aorta is much, much smaller than the total surface area that can flow through in the entire network of capillaries at some distance from the heart, okay? There's billions of tiny capillaries that blood vessels can flow through. So you have to think of the entire cross-sectional area of that entire capillary network that blood can flow through, okay? So you're talking about, thousands of times greater area that can flow through those capillary networks. And that is why the blood slows down so much in the capillaries, despite this relationship here. Okay, in any individual given plate. Now, we also have, of course, in order to get flow, we have to have some area that material can flow through, but also you have to have some pressure difference. Okay, and so pressure is going that pressure difference is achieved by the heart. This is the whole entire purpose of the heart, is going to pump and increase the pressure on one end of the circulation. And here we see the blood pressure changes in the systemic circulation, okay, during ventricular systole at the peak and diastole at the sort of base here. This tends to vary between about 120 millimeters of mercury to 70 millimeters of mercury in a healthy adult. That can change with hypertension. That might be defined, sort of borderline hypertension might be defined as 140 millimeters of mercury during systole, and sort of hypertension where you actually need to get treatment and take statins and things like that, going up above 150 millimeters of mercury. Of course, when you have higher blood pressure, that can lead to risks and damage to the vessels of your circulatory system, right? The higher the pressure it is, the more likelihood you have for a hyped brain, okay? Much like the more likely you have of a chance for a blood vessel to rupture. What do we call a blood vessel that ruptures spontaneously? It's a stroke, right? So you have a ischemic stroke. Okay, you can have a blood vessel that ruptures. You can have a bleed. If that happens in a place like your brain, a brain aneurysm, it can be very damaging to the health of that surrounding tissue. Because one, you've got a bunch of now buildup of blood and fluid that's not supposed to be there in some area of the brain. And two, you've now cut off the oxygen supply to things immediately downstream of that blood vessel. And that can lead to cell death and loss of function. Okay, so what else do we need to talk about on this slide, right? So the pressure changes in the sort of artery side have pulses, okay, due to the contraction and relaxation of the heart. It never gets down to zero here, though, because of that elasticity of the arteries and the artery walls that provide some resistance and energy that can snap back and spring back into action when the heart is relaxed. Now, as we go through the circulatory system, we can see is that there is, in the capillary beds, there is maybe, you know, at the upstream end of the capillary bed, maybe there's like 40 millimeters of mercury. By the time you get to the downstream end, maybe it's 15 millimeters of mercury. Okay, so there is a pressure gradient through those capillary networks, and which is what's going to cause continuing flow through those capillary beds. Does anybody know what millimeter of mercury is? Like that, why do we even use this term, right? It's like a very old school way of how people used to measure pressure, okay, which would be to take a long tube full of mercury that you'd fill all the way up, put your thumb on it, and then put it upside down in a pool of mercury, okay? And then the mercury would sort of fall to a particular level, okay, which would create a vacuum on the top, and the surrounding air around you, okay, would be pushing on the pool, pushing the mercury back up. And that would be how high that column of mercury is, okay, is determined by how much pressure in the air above us is pushing down on that pool. Okay. That's usually about 15 pounds per every square inch of this fluid on top of us pushing down on that pool, or about I think about 1,000 millimeters of mercury, about 1,000 millimeters of mercury will be pushed in a normal atmospheric condition. Okay, so this pressure change here is actually relative to the overall surrounding pressure that we're in. Okay, we're going to just say we're always sort of surrounded by air. It's always pushing down on us on all sides. So, but within our own blood vessels, we have an additional buildup of blood pressure that will allow material to move. And so we can measure our overall heart rate, like the classic way that you would measure heart rate is with a stethoscope. Okay, and how would it actually measure the blood pressure is also with a stethoscope. Today, they do this usually digitally. You stick your arm in a sleeve and it does something to you, squeezes your brachial artery, and then somehow calculates your your blood pressure. How do you do it actually as a doctor? If you're using a stethoscope, and what are the actual triggers that we use to identify that blood pressure? So if you wrap the bicep and the arm with this, with an air cuff, okay, you can then listen to blood flow at the output of this brachial artery and listen to pulses of pressure changes as blood sort of passes through this artery. Now, what happens is, of course, we all are surrounded by some level of pressure from the air, okay? But if we increase that pressure selectively at one location of our body, that's going to tend to block off that blood and collapse the artery at some point, right? If you just push on it lightly, it's not sufficient amount of pressure to overcome the internal blood pressure in that artery. But as we squeeze harder and harder and harder, eventually we cut that pressure off, okay? So, but here's the thing. When you have a heartbeat in your, when your heart is beating, right, and you're going through these cycles of systole and diastole, and the artery is fairly close to your heart, then you have these pulses of pressure, right? As at some level, if the pressure that you are applying to the artery is greater than every moment of the cycle, you'll completely block off the flow of blood here, right? So if we inflate the cuff, okay, up to, I don't know, 200 millimeters of mercury, okay, what will happen is there will be absolutely no blood flow through here. You've completely clamped down on the artery. If you're listening for that blood flow here, okay, you'll hear nothing. Okay. Now, if you release the air pressure slowly, okay, at a certain point, the blood pressure inside the artery will be sufficient to overcome the extra external pressure on that muscle and on that artery. And at that point, you'll get a pulse of blood flow through the artery. But that will only happen at periods of time in which the pressure is higher than the external additional pressure. So in the range between the peak of the systolic pressure and the bottom of the diastolic pressure, you'll get transient flow of blood through that artery. Okay, so you hear these little pulses of blood coming through. Okay, and as you and the moment, the level at which you first hear those pulses is going to be the peak of that systolic pressure, right? So above that, you'll hear nothing. As you just start to dip below that, that's when you'll first start to hear those pulses. Okay? So that first moment that you hear that, that's defined as your systolic pressure. As you then continue to drop the blood, drop the cuff pressure, at some point, you're no longer going to have transient intermittent flow. You're going to have continuous flow. Because if you drop the pressure below the bottom of the diastolic pressure, then blood will always be able to overcome the resistance to flow from the cuff. Okay, so now that sound, that pulsing sound will go completely away. Okay, you will just be able to hear sort of the rush of blood through the artery. Okay, so when that sound goes away, that is going to be your diastolic pressure as you continue to relax it. Okay, now blood pressure is also sort of dependent, at least in large animals, is actually dependent on body position and body size. Okay, the blood pressure I need to maintain blood going to my head is about 30 millimeters of mercury more when I'm standing up than when I'm laying down. Okay, that's because I have to push the blood up and resist gravity to send it back up to my head. You have to have a higher level of pressure. The bigger you are and the more vertical distance you have. So, for example, if you have a sudden drop in blood pressure, a common sort of reaction to that is fainting. You fall on the ground. And why is that sort of an evolutionarily positive thing to do, putting yourself in a very vulnerable position? It's because you need to restore the blood flow to your brain. And by getting in a prone position, you need 30 millimeters of mercury, less blood flow to continue that circulation. Because now you don't have to resist gravity. And you can see, you might imagine, like, what about like a really tall creature like a giraffe? Giraffes have very powerful hearts. They're pumping at 250 millimeters of mercury because they have to overcome all of that resistance from gravity to flow up to the brain. Okay, so let's take a look now at capillary blood flow with an actual video of what this blood flow looks like. And what we can see here is a variety of different blood vessels, including capillary networks. So this is a collecting venule, one of the first collecting venules, the first stages where blood flow is going to, where the red blood cells are going to sort of leave the capillary beds and return to the venous circulation. And what do you see here? This is microscopic zooms here, and you can see these little red blood cells slowly creeping along, right? This is very slow, right? Think about this, like the width of red blood cells, maybe 10 microns wide, and this is moving like, maybe 20, 30 microns per second. It's really slow. That's giving you the red blood cell plenty of time to exchange gas with its surroundings, okay? If you look closely at it, you can see that the red blood cells almost look like little hot dogs, okay? So red blood cell is actually sort of like a flat disc that's a little bit concave, biconcave on both sides, when it's just sitting around in a big free solution. But if you put it in a capillary, the width of capillaries inside a lumen of capillaries is actually a little bit more narrow than the diameter of a red blood cell. So that's going to squeeze it into little tubes, little hot dogs traveling through tubes, which is going to then further decrease the distance between the inside of the red blood cell itself and the outside of the capillary. And as you can see, right, as soon as the blood cells get into this venule, you see a sudden acceleration, right? So the wider your tube, right here, as they get into this, the resistance and the friction that this capillary network is sort of producing to slow these red blood cells down, goes down, that once you are able to sort of pull yourself away from the sides of the capillary wall. Now, we have a whole bunch of capillary beds within our body, and at any given time, only a small fraction of them are open, okay? If they were all open at the same time, you'd go into shock, your blood pressure would fall dramatically because you'd have way too much surface area that blood could flow through, okay? So we have to regulate the ability of blood to enter capillaries, okay? And there's sort of the primary two ways that we do that is through the arterial smooth muscle. So these are the smooth muscle that can vasoconstrict, that is to tighten down the diameter of the arteriole, or vasodilate, which is to open up and relax and increase the diameter of the arteriole. And we also have something called precapillary sphincters, which are little rings of muscle that are at the entrance to each little capillary bed, okay? Those two can either dilate or constrict. And by doing this dilation and constriction in a targeted and controlled manner, you can now shunt or move blood to different parts of the body that need it more and prevent it from moving into areas that don't need it. Okay, so this is going to generally depend on the metabolic load of the region that the blood is traveling to, right? So if you're burning a lot of ATP, if you're burning a lot of glucose in a particular area, you have a high metabolism in the area, maybe because you're running, let's say, then your legs are going to have dilation of your precapillary sphincters. The arterioles will have vasodilation that feed into those capillary beds as well. And so you have a lot of blood flow that gets directed to the leg muscles, okay? On the other hand, if you're relaxing, okay, you're going to vasoconstrict those arterioles that go to the legs or to your other muscles, and you'll dilate the arterioles that go to your gut, where digestion can take place. So the control of this dilation and constriction and where that dilation and constriction takes place is really regulated by both your parasympathetic and sympathetic nervous systems, okay? They can both direct electrical signals to open and close particular smooth muscles, as well as release particular hormones that will cause a more systemic dilation or constriction. So, for example, epinephrine is a potent modulator of constriction, okay? It causes constriction in areas that are used for digestion. Okay, so you're going to shunt, if you have a sort of jolt of epinephrine, you're going to shunt blood away from your gut into digestive systems, and it will cause a vasodilation of muscle areas. Okay, so you're going to reroute blood to muscular areas so that you can run away. Okay, now here's an example now of vasoconstriction and dilation. And in this example, we're going to see an example of an arteriole that was previously sort of constricted via norepinephrine. Okay, so this must be one that's going to the gut. Okay, and when we apply this other sort of compound adenosine, which will cause a vasodilation, we can see a tremendous change in both the diameter of this arteriole, okay, which is increasing by, I don't know, a factor of, diameter of probably 3, and an overall cross-sectional surface area of about 10 then. And what we see also is this sudden sort of flooding increase of delivery of blood to the surrounding capillary networks. Okay, so now correspondingly with this constriction and dilation, that also is often going to be paired with changes in the overall level of blood pressure and heart rate, right? As we increase our cardiac output, you need to have some vasodilation in order to prevent your blood pressure from spiking to hundreds and hundreds of millimeters of mercury, right? If you increase your cardiac output can go from sort of 5 liters per minute to 30 liters per minute. And our blood pressure really is going to change in sort of much smaller ranges. Okay, it'll go up a little bit during exercise, but the vasodilation, that increase in cardiac output is going to be paired with vasodilation to try to maintain a similar level of blood pressure throughout that entire exercise sequence. Okay, so now what actually happens in these capillary networks? How does gas exchange, and how does fluid flow within these capillary networks? So we know, right, that there's a pressure gradient between sort of the input area to the capillary bed and the output area of the capillary bed, because otherwise there wouldn't be any fluid flow through the capillary, right? So there has to be some sort of pressure gradient, a pressure drop. And what pressure I mean by this is what's known as hydrostatic pressure. Okay, so this is the pressure that is produced by the elastic forces, the constriction of the heart, and the elastic forces along the arteries. Okay, so it's pushing down on the fluid within the lumen of the capillaries. Okay, now there is, and what that hydrostatic pressure will tend to do is it's going to cause fluid to leak out of the capillary. So there's no fluid that leaks out of the arteries. Okay, so they're thick. Okay, they've got these really thick walls. They've got multiple rings of stuff. Once you get into the capillary beds, though, you only have a single cell layer of endothelial cells in that little wrapper basement membrane around it. Okay, so it's very thin, and actually there can be gaps, microscopic gaps between each one of those endothelial cells. Okay, that can allow fluid to transport out. Okay, and the greater the blood pressure, or the greater the hydrostatic pressure here within the capillary, the greater the rate of that fluid leakage will be. Okay, because the overall pressure in the interstitial fluid is pretty much constant, or hydrostatic pressure is pretty much constant. We have a higher hydrostatic pressure within the lumen of the blood vessel here, then you're going to get net movement. We're going to get some fluid flowing out. Now, the red blood cells, which are the primary constituents of the blood, or the primary cellular components of blood, are too big to fit through those crevices. Okay, so they have to be retained. And in fact, those crevices are pretty small, so narrow that there's also many other non-cellular components of blood that, like large proteins, that also cannot escape. Okay, so those proteins form a colloid, which is basically sort of a bunch of stuff floating around within a fluid. Okay, and that colloid pressure produces another force that tries to bring fluid back into the within the blood vessel. And so, and why is that? It's like, it's sort of like, it goes back to like the second law of thermodynamics, which basically states that disorder is always going to increase in a closed system. Okay, so to increase the disorder in a system, we want to have an equal concentration of stuff dissolved on the inside and the outside. Okay. Now, if there's more stuff dissolved or in a colloid on the inside of this blood vessel, then water is going to want to try to flow into here to balance that level of concentration on the inside versus on the outside. So this is called osmotic pressure. Water is always going to try to flow from an area of low concentration of stuff dissolved solutes to an area of high concentration of solutes if there is a passageway that it can travel on. Okay, that's called osmosis, right? So that osmotic pressure is continuous actually along this entire length of lumen, this pressure between the inside and outside of the cell. Okay, and it's really due to these concentration differences of the proteins dissolved inside and outside of the lumen. So if that pressure is constant, okay, this osmotic pressure is constant, and the blood pressure has to be dropping as you move through the capillary, you might imagine that there will be different amounts of fluid leaking out at different locations along the length of this capillary bed. And that's correct. In fact, at a certain point down the capillary bed, the net flow of fluid actually reverses direction. Okay, so at the sort of input side of the arterial end of the capillary bed, the blood pressure, the hydrostatic pressure is greater than the osmotic, the hydrostatic pressure pushing out is greater than the osmotic pressure pulling the water back in. But by the time we get to the venular end, okay, the hydrostatic pressure is no longer sufficient to overcome the osmotic pressure of water pulling back in, and we get a return of water back into the capillary. Okay. So, and this is really because the sort of pores here between the endothelial cells are only big enough to allow water and some very small solutes like ions to pass out. It won't allow large proteins like albumin, which is a major component of blood, to leak out. The thing is, okay, so we have this constant sort of bulk flow of fluid out and into the capillaries, as well as a containment of the major protein and cellular components of the blood within the capillary itself. Simultaneously, we're having gas exchange with oxygen, sort of unbinding from hemoglobin here and diffusing out into the interstitial fluid. From there, it can diffuse into the tissues of the body and deliver it to those body tissues. Meanwhile, within the body tissues and the cells here that are undergoing some metabolic activity, carbon dioxide is being produced and it's coming out to here and diffusing and equalizing its concentration within the interstitial fluid and eventually into the capillaries here. And that will get either dissolved within the capillary itself, within the blood itself, or binds to hemoglobin and get carried up to the heart or up to the pulmonary circulation. Yeah.
15% forced out of the capillaries, like the blood.
So blood is not forced out of the capillaries, but water is, and water and very small solutes can get forced out from that pressure. Okay? And much of it comes back in by the end, but not all of it. About 15% of the cells does not make it back into the capillary. The drop in hydrostatic pressure is not sufficient to fully return all of that fluid to the capillaries. So where does that fluid go? Okay, then we're basically what I just told you is our capillaries are kind of leaky, and we're leaking like fluid into the sort of tissues surrounding our body all the time. And that fluid still needs to get returned into the blood somehow, right? So that is we have a whole nother parallel circulatory system, which is not driven by a heart and pressure, which is called the lymphatic system. And this is basically, you could think of it like the sewer pipes or something that goes down and returns all of the leaked out fluid back into the circulation over much slower time scales. We only need to have about 4 liters of fluid per day to get returned. But these sort of lymphatic vessels are all over the body. Okay. Furthermore, there are little valves within the lymphatic system that will cause the fluid to flow in one direction only, right? Because, and the lymphatic system is basically driven by contractions of muscles. If you can contract a muscle in one location, you're going to push fluid. You're going to squeeze the fluid out of that particular area of lymphatic vessel. If there's a valve that allows it only to flow one way, then it'll tend to flow in that direction. It won't backflow. Those lymphatic vessels will pool in a spatially distributed set of nodes called lymph nodes. We'll go into those in a lot more detail in the immune system, because those are the site of a primary site of where your immune system is going to screen your body and the fluids within your body for pathogens. Okay. You can imagine that anything that sort of gets into your blood and it creeps out, maybe some bacteria, some viruses, something that have creeped out and have infected some part of your body tissue. And now those bacteria are sort of multiplying, they're in your interstitial fluid. They're going to be then transported from your lymphatic system to these lymph nodes, where they will be recognized by white blood cells and responsibly coordinated against them. And so, you can see you'll have a migration of white blood cells to lymph nodes to destroy the pathogens. And this is why when, if you have an illness, okay, then lymph nodes that are near in location to where the battle against that pathogen is taking place will tend to swell up with pus, with migration of these cells. So, if you have a head cold, then you can feel lymph nodes right here, okay, become larger and swollen. If you have cancer, okay, the cancer has metastasized and spreading, then those cancer cells will first be found in the lymph nodes that are surrounding the cancer site. And that is the dissection and biopsy of lymph nodes is a common common practice for trying to stage a particular cancer. Are you stage 1 if it hasn't expanded outside of its little area? Stage 3 or stage 4 generally indicates that the surrounding lymph nodes have been invaded, or other parts of the body have been invaded by that cancer. Any questions on this stuff so far? Yeah. Right. How does it get other resources like glucose? Yeah, so glucose can actually leak out if there's food is small enough. Also, there are glucose transporters that will bind to glucose and transport it across cell membranes effectively. So glucose transporters are many of our nutrients that we have that we need to get concentrate in one area versus another area will have specialized sort of protein-based transporters that will spend ATP to move the glucose from one end to another end of a membrane. Okay, and we'll see that in transport epithelia most specifically. And I think after spring break, get into it. Any other questions? Okay, cool. Okay, so let's take a look here. Here is a set of 2 X-rays, right? And let's sort of stare at them. Pre-radiology here. What do we see about these 2 X-rays? And what is even an X-ray? An X-ray is like a high energy particle that can pass through tissue pretty easily, but it can get deflected and blocked if the tissue is of a higher density. Okay, so areas that have and what we see here is basically a negative image of the areas that allow transmission and don't allow transmission. So dark areas are allowing transmission of an X-ray through it, and the white areas are places where the X-ray has gotten blocked. Okay. So these are our lungs. Okay. And what we see here, so the lungs are mostly air. So the X-ray goes easily through those. But then on this side, it seems like we've got a little bit more white stuff in these lungs, right? So that could indicate that there is fluid building up. Secondly, we see this sort of bulge here. This is the heart. Okay? And we see an increased size of the heart in this one. Okay? So these two conditions are linked together, and this is an X-ray of a healthy person and someone who has congestive heart failure. Okay? The congestive heart failure is characterized by an enlarged left ventricle and left chambers of the heart and seepage of fluid into the lungs, which is called edema. So how does this take place? So congestive heart failure can be triggered by a variety of factors, and it's generally a progressive disease. Okay? And it is going to oftentimes be caused by a weakening of the left side of the heart. So the left side of the heart, remember, is generally the stronger side. It has a thicker muscle, and it has to have that because it has to pump the blood to a much higher pressure to get it through the systemic circulation than the right side of the heart, which only has to pump the blood up into the pulmonary circulation, which has much less resistance. Okay, so the left ventricle could become weak. How could it become weak? Well, you might have a reduced amount of blood flow to that area, so you're starving of oxygen. Maybe because you have arteriosclerosis, so a buildup of fat or plaque deposits within the coronary arteries that feed the heart muscle. Okay? And so you have sort of reduced blood flow, reduced activity, or reduced sort of delivery of glucose and oxygen to that part of the heart that could make it weaker. Okay? As that ventricle weakens, okay, it's not going to be able to pump as hard. It won't be able to compress as hard, and it'll reduce the overall amount of blood, cerebral volume that can get pushed out into the systemic circulation. Okay? So if that happens, though, the right side of the heart is, it doesn't have to work as hard anyway. Okay, if that right side of the heart is not weakened, then what will happen is there'll be an imbalance in the cardiac output of the right side versus the left side of the heart. Okay, so if the cardiac output of the left side of the heart is sort of falling and the right side of the cardiac output on the right side wants to stay the same, there's going to be an imbalance. That is going to cause cardiac output in this sort of the right side to sort of shoot up into the pulmonary circulation, okay? And it's going to apply an inappropriate amount of pressure to this area. Okay? This area, since we have not expelled enough blood volume from the left side, okay, it's going to sort of cause a backup and an increase in back pressure into the pulmonary circuit, okay, because it cannot clear, it cannot fully expel this fluid, so you're going to need more pressure here to get blood into the left ventricle. That is going to cause an increase in blood pressure in the lungs, and what we just saw, right, with those capillary networks, right, as we have higher hydrostatic pressure, where you have more fluid leakage out. So if we have an increase in hydrostatic pressure in the capillary networks, we're not going to be able to provide enough drop in that hydrostatic pressure relative to the osmotic pressure to return fluid back into the capillaries by the end of the pulmonary circulation. Where is that pressure going to go, or where's that fluid going to go? It's going to leak out into the lungs. That's then going to cause difficulty, sort of a degrading of the ability to do gas exchange within the lungs. And that will cause the left ventricle to have to pump harder, okay? Because we're not getting sufficient gas exchange. We're not expelling enough carbon dioxide. We're not getting enough oxygen in. And it causes a negative feedback loop where the heart has to work harder and harder and harder to achieve the same overall outcome of how much gas is being delivered to the rest of the body, because, and which will then tend to sort of weaken the left heart more and more. So this can cause sort of a downward spiral and lead eventually to death. Yeah. Lead it to be what? Be car?
Big part.
A big heart. Oh, good idea. So how does it lead to a big heart? Okay, so if this ventricle is weak, okay, then this ventricle is still pumping. Okay, we have an increased blood pressure here. That increased blood pressure here is going to maintain a higher blood pressure in the heart itself during relaxation. That's going to tend to stretch out that heart over time. Okay, and continue to, if you're always pushing, if you can't get down to sort of zero blood pressure on the left-hand side, you're constantly having too much pressure on the left ventricle and it's going to swell. Yeah, it'll take time. I mean, we're talking, this is a process like congestive heart failure can last for years, right? This is sort of a leading cause of death. So it doesn't happen overnight. Other questions? Okay, so we've talked a little bit about like how the blood is getting around. Okay, what is that blood actually composed of? Okay, and we've got about 5 to 8 liters of blood within our body. About half of that is known as plasma, and half of that is the cellular elements of blood. Okay, so what is the primary component of plasma? It's just water. Okay, but then we have a whole bunch of other ions. Okay, that we need that we need in order for the electrical properties of our body to function appropriately. Okay, so we have sodium and potassium, calcium, magnesium, chloride, bicarbonate, protons, H+. Okay, so these are like the key ions that you really will need to know. So sodium and potassium, why are they important? Okay, sodium and potassium are the primary ions that drive what's known as an action potential. Okay, this is how electrical currents are transmitted along your nerves. They're how electrical signals within your heart across your cardiac myocytes occur. Sodium causes an increase in the sort of electrical potential across the cell membrane. The potassium tends to bring it back down. So the rapid opening and closing of sodium and potassium channels is what causes pulses of electrical activity, both within your heart and within your brain. We have calcium. Calcium is the #1 most important intracellular second messenger in your body. Okay? In every cell, if there was ever a guess of what second messenger is involved in the process, I would guess calcium. And this is because calcium levels are held at extraordinarily low levels within the cytosol of your cells, okay? But are at relatively high levels outside the cell and within places like the endoplasmic reticulum. So if you open a channel that allows calcium to pass into the cell, you'll have a huge increase in the relative concentration of calcium. Okay? And that can cause a very potent signal to many other intracellular proteins and organelles. Okay? So calcium is a very important signaling molecule. Magnesium is a regulator of other channels that can selectively block them and allow calcium to flow in only at certain times. Chloride is an ion that is involved in suppressing the electrical changes within the electrically active cells within your body. Bicarbonate and H+, the proton, these two things are the main sort of buffers of pH within, or I guess H+ is not a buffer, but so pH is a measurement of the concentration of protons within the body. How acidic is your body tissues or is your blood? And then bicarbonate, Hc3- is a buffer that can bind onto the protons or the hydrogen ions and keep them within a narrow range of pH. So certain sort of intracellular processes will only operate appropriately at a particular pH range. So if you have too much acidity within your blood, a whole host of negative consequences can take place. Okay, so also within our plasma, we have proteins. The #1 most abundant, many different proteins are floating around in our plasma. The #1 most abundant protein is called albumin. Okay, and this is a protein that essentially exists to sort of shuttle other proteins around that are hydrophobic. So any protein or any sort of molecule can have hydrophilic component of it where it likes to get dissolved, in water, because it has some polar nature to it. And then you have nonpolar molecules that are hydrophobic. Did I say that wrong? Hydrophobic is nonpolar. Okay, hydrophilic is polar. Okay, albumin allows hydrophobic molecules to bind on and latch on to hydrophobic locations within the albumin and makes them now soluble within blood. And that allows them to transport these different proteins around. And there's tons of proteins that you need to get delivered in different places. Albumin is the sort of primary transporter there. And by transporter, I don't mean across a membrane. I mean like a carrier that allows it to travel and circulate throughout the blood. Yes. Okay, so hydrophobic molecules can bind onto albumin, which is a large sort of protein that floats around in the blood at very high concentrations to any other protein in the blood. That protein is also so big that it cannot leak out of the capillaries and is the main driver of that osmotic pressure. Then we have nutrients like glucose, right? Or other amino acids that are going to be traveling through that can be turned into proteins. We have wastes. Those are metabolic byproducts, things like carbon dioxide, like lactic acid, urea, things that are metabolic breakdown products that we need to get rid of. And then we have our gases, okay? Oxygen and carbon dioxide are going to be, I mean, nitrogen's also there as well, but we don't talk about nitrogen because it doesn't really do anything in the body. But the oxygen and the carbon dioxide are the sort of main drivers and things that we need to keep in balance with one another. They're sort of at opposition to each other, oxygen and carbon dioxide. Carbon dioxide will interact and be converted into bicarbonate through an enzyme we'll talk about in a little bit. And then finally, we have hormones. Okay, hormones can be small molecules or they can be proteins. They are things that are dissolved into the blood and travel to send a signal to another part of the body, right? We can send signals to other parts of the body through electrical signals, like from our brain to some nerve that goes down to a part of our body, vagus nerve, maybe. And that's a fast way of transporting stuff there. Okay. But that can only target locations that have a nerve that goes to it. Whereas hormones can get produced by a different part of any sort of organ in the body that makes hormones, or any tissue that can make hormones, and they can sort of release those hormones into the blood, which will then bind onto receptors at any location where they are found within the body and allow coordination of activity across different organ systems. The hormones are basically messenger molecules. So that's the plasma. It's about half of it, right? Then we've got our other half. What are our other half? We're going to call them cellular elements, okay? Because they are from cells. They are either cells or they are derived from cells. The 99% of them are going to be erythrocytes, which are red blood cells. Erythrocytes are cells that have no nucleus, okay? They're enucleated, okay? They are essentially just bags full of hemoglobin, okay? They are going to be primary oxygen transporters. They also will transport carbon dioxide. They're like a supplemental transporter for carbon dioxide, but they are the primary transporter, or sort of carrier, I should say again, of oxygen. And then we have the leukocytes. These are white blood cells. There's 7 major classes, but there's many subclasses of these. And these are like literally like 1,000 times less prevalent than the erythrocytes, but they're absolutely important for making sure our bodies do not get invaded and taken over by pathogens, right? And then finally, we have platelets. So platelets are derived from a cells called a megakaryocyte, which basically then breaks up into little fragments, and the platelets can travel around and are absolutely essential for blood clotting, which will prevent blood from seeping out if you cut and damage a blood vessel. Okay. We can sort of, map these. These are all derived from what is known as a pluripotent stem cell, one originating progenitor cell within the red bone marrow, which is the material inside the hollow spaces inside our bones, which is the origin of all of these cells. Okay, so we have some stem cells there that can self-renew, and they can continue to make copies of themselves over your entire lifespan, or they can also differentiate into other classes of stem cell, lymphoid stem cells or myeloid stem cells. These are differentiated by where the cells will mature, either in the lymph nodes or lymph fluid or in the bone marrow itself. And then these stem cells can also self-renew or transform and differentiate into a large variety of different cells that we'll sort of go into the details of each one of these cells in the immune system. Okay. Sort of most importantly for this particular circumstance here, this particular slide, these myeloid stem cells are what can turn into erythrocytes, a process called erythropoiesis, which is regulated by erythropoietin which is something that can increase or make more potency of the production of erythrocytes, red blood cells. Yes. Very good. Could you speak up a little bit? Of Mg2 plus. So the significance of Mg2 plus, so that's the magnesium ion. So the magnesium is an ion that really functions as a regulator of particular ion channels. There's, and we'll cover it in more detail in the nervous system, but there's ion channels that will open and close depending on the voltage difference in a cell membrane. Okay, and they can also be plugged or regulated by the presence or absence of magnesium, therefore, because magnesium is an ion. You can imagine it like this. Like imagine you have like an ion channel, right? And it can open up, and that opens up a pore that allows certain ions to flow through. Okay. Magnesium is a positively charged ion, okay, but it has a different sort of shape to it. It's a little bit bigger, okay, and so it can get into the pore, but then get stuck. So on the electrical charge, magnesium on the outside of the cell, okay, if you have a large, if the inside of the cell is very negative, it wants to go into the cell. It can try to get into the hole, the pore of that, but then it gets stuck in it because it can't fit all the way through. So there can be a regulator of electrical signals through various ion channels. So it's quite an important molecule, even quite an important ion, even if it's not as primary to the establishment of membrane potential and the changes of membrane potential. Okay. Okay, so we've got, okay, so in terms of these classes, so we have erythrocytes. Their development is sort of enhanced by erythropoietin. We also can create these platelets, which are circulating throughout the body and regulate blood clotting. And then we have a range of these different white blood cells here, okay? And we've got basophils, eosinophils. Each one of these has a sort of different sort of function. The ones with stars are phagocytes. So these are cells that can phagocytose pathogens. Phagocytosis is a process of essentially surrounding and eating and dissolving, but degrading, breaking down any pathogen. And certain phagocytes, like the neutrophils or macrophages, these will serve to primarily just seek and destroy these sort of pathogens, whereas other phagocytes can take what is broken down and display it on their surface in order to trigger and coordinate a greater response against the sort of molecular components of that pathogen, and train B cells and T cells to go out and specifically identify and attack those types of pathogens. It's a process in the active immune system, which we'll go into after break. Okay. Okay. So the blood is, incredibly important, right? We have to have it. We have this vast network of blood vessels within our body. Now, problem is sometimes things can go wrong. If you have a cut, right, and you cut either a vein or an artery or arterial, you have to have some mechanism of stopping the bleeding. Okay. And so circulating within our blood, we have a variety of components here. And we have, in particular, we have platelets that are circulating, and the platelets will maintain their sort of ability to not stick to things for a period of time, but then through a cascade of factors, okay, that can be triggered by the release of the presence of collagen within the blood itself. So if you puncture like a blood vessel, that will expose this outer layer of membrane that contains collagen that wraps it to the intracellular components of blood, or the intraluminal components of blood, sorry. That can trigger a cascade that then causes a network of fibers to form. Okay, and those network of fibers that form are specifically sticky to platelets. Okay, and so circulating, we have this soluble protein called sort of prothrombin that when this enzymatic cascade, and I'm not going to tell you the cascade because it's like 15 steps long, and you'll probably never need to know it. But there's this cascade where that can be triggered by the presence of these collagen fibers. They can detect it. It causes all these sort of chain of events to occur, which then will convert this sort of enzyme from prothrombin to a thrombin form, which then causes the positive feedback loop that tends to convert any other prothrombin around back to thrombin. And this thrombin enzyme itself will catalyze the conversion of fibrinogen to fibrin. So fibrinogen is a molecule that is the genesis or the progenitor of fibrin, and it is a soluble molecule. When it gets converted to fibrin, it sort of falls out of solution, becomes sticky, and sticks to other fibrins. Okay, so in the location where we have a high concentration of these factors that drive clotting from the damage around this area, we'll have a specific conversion of fibrinogen to fibrin and a formation of a fibrin clot. Okay, that is going to cause platelets to stick to that plug, okay, and basically occlude the hole. Okay, that will prevent red blood cells from leaking out. As it gets thicker, it's going to prevent fluid from leaking out, and that will literally stop the bleeding. Okay, so hemophiliacs are people that have a mutation in some part of this cascade. There's many different sort of forms. And the end result of that is that they cannot catalyze the conversion of prothrombin and thrombin, fibrinogen to fibrin, and they cannot stop the bleeding without sort of external factors and medical attention. Okay, any questions about this? Yes. So the clot will eventually get broken down by other white blood cells and components. Once you know, there's going to be some immune response that then sort of cleans up all of the damage around it through a variety of different leukocytes. Yeah. They are proteins that are sort of produced within. I'm not sure what the origin of them, what cellular origin it is. I'm guessing probably the bone marrow. And they're just going to be constantly produced and sent out around our blood. Okay, so it's constantly in our blood. Okay. Okay. Well, we could we might as well get. Oh, I can do a little bit like, let's do 5 minutes on the respiratory system, just to introduce you to one component, and then we're going to take off a little bit early so the next class can come in here for an exam. But let's take a look at this. So let's look at the respiration system. So here's another example. Okay, this is an example of what's known as ground glass opacities, looking at a scan of a person's lungs. What we see here is that there's these like strong sort of lesions within the lungs, which break down, which prevent gas exchange and prevent air from going into and out of the lungs. This is images of sort of an advanced progression of COVID in sort of the first months that COVID came out, the original sort of form of it. Okay, so we need to have the respiratory system sort of requires a variety of different concepts. Now, first, we need to understand partial pressure in gases and liquids. How is the pressure that is all around us translated into the pressures of individual molecules like oxygen, nitrogen, carbon dioxide? How does that influence how much material can get dissolved into a fluid and how that can get transported to different locations? Then we have what's known as a countercurrent exchange, which is a method of overall system design, which is going to optimize the efficiency of exchange across two different circulating systems. That's going to inform the structure and the function of the lungs. And we also want to understand how carbon dioxide and oxygen are sort of in opposition to one another, and how that relates to blood pH, because carbon dioxide has a strong influence on blood pH due to the enzymatic conversion of carbon dioxide to bicarbonate. And then finally, how are these carbon dioxide and oxygen going to sort of bind to get transported by and unbound from hemoglobin? I think I'm just going to stop here because I think just getting into this is going to be too much. So we'll go into we'll get all the way through the respiratory system on Thursday. Thank you so much for your attention and good luck.